And of course, by the way, you're welcome to chime in when we talk. All right. So first thing first, blood. Okay. So for the blood, you have to understand what it does in terms of the homeostasis maintenance. So transport, right? The functions first. The transport, the electrolyte balance, protection, white blood cells, temperature regulation, and all that listed. Characteristics and quantity we talked about it. Blood, you have about 5.5 liters, something like that. Depends on the body size. Characteristics pretty much links with this, the composition of the blood. Okay, that it's a fluid essentially. Fluid, it's a connective tissue. Now, composition. Understand what are the components of plasma. Okay, you have to recognize things like that. If I will ask you, red blood cells, are they components of plasma? Obviously not. That makes sense? Formed elements. Know all the blood cells. It's not a big deal, I suppose, knowing all blood cells, right? Red blood cells, RBCs. Structure and function, and understand how structure is linked to function. Remember we talked that erythrocytes are pretty compressible, so they can squeeze through the capillaries. Why they have increased surface? Why they have a shape like they biconcave? Increased surface, increased rate of gas exchange, right? More surface, faster exchange. Leukocytes. I give you the list here of what you should know. Okay, granulocytes, agranulocytes. For each function, of course, is the main one. Okay, know what they protect for. If I will ask you monocytes, do they phagocytose? What would you say? Monocytes? No, they have to become macrophages first, right? Platelets, they're pretty much pockets of cytoplasm that pinch off the megakaryocytes, and that they function not only regulation of clotting, not only the formation of the platelet plug, but also, remember, they regulate what you might call it inflammation. Does that make sense? Remember, it's quite important. Hemostasis. I highlighted the steps here. So understand, for example, the difference between the platelet plug and the clotting. So platelet plug is when platelets become activated, right? And they start to stick to each other. Does that make sense? So activated platelets attach to the surface of the endothelial cells, blocking the injury, plugging the injury. They release chemicals that activate other platelets. Activated platelets, it's positive feedback loop. More and more activated platelets pile up. Without clotting, will that plug be stable? No. So that plug will be unstable unless clotting factors via intrinsic and extrinsic pathways, right, will stimulate clotting, will lead to formation of the fibrin mesh. So for the clotting, know the pathway. Especially focus on the common pathway, okay? That starts with prothrombin activator that converts prothrombin into the thrombin. So understand that, what are the stimuli for intrinsic and extrinsic stimuli. Rate, which one's faster, which one's slower. Does that make sense to you? Perfect. Understand, we mentioned some anti-clotting medications, aspirin, heparin, warfarin, dabigatran. Just have an idea what those words are. Blood groups. Understand that. Okay. Like if I tell you, well, I may ask you, blood group such and such. Say, blood group A, negative. What does that mean? It has, what does that mean? It has A antigen. It has antibodies to B. And if it's rhesus negative, does it have antibodies to rhesus? No. They're not pre-existing. They're formed after the exposure. Make sense? Know that. These are the conditions that we discussed. Understand what they are. Sickle cell anemia and thalassemia associated with RBC formation, right? The improper hemoglobin, okay? Leukemia, white blood cells. Hemophilia, clotting. Thrombocytopenia, platelets. Okay? We haven't discussed the blood tests. No worries. Immune system. Understand the first line of defense. What would be the first line of defense in the immune system? Very, very first. Skin, mucosa, right? Different chemicals that are produced with the secretions. Right? Make sense? So understand that, the difference. Okay, phagocytes, mechanisms. Yeah, immune cells. So what do you have to know about immune cells? Going back to this, okay? So know the cells of an innate immune system. Can you tell me those now? Neutrophils, basophils, eosinophils, monocytes/macrophages, and NK lymphocytes, natural killer cells that fairly non-specifically destroy cancerous and virus-infected cells. Yes.
So are white blood cells different than immune cells?
No. White blood cells are immune cells, essentially. So it kind of goes again about white blood cells. Essentially this, and, this is similar, okay? It's kind of repercussions. But what I absolutely want you to know is what are the mechanisms of defense for those white blood cells. Like neutrophils phagocytose, basophils don't. Macrophages phagocytose, lymphocytes don't. Instead, lymphocytes induce apoptosis. Okay? What these cells, what do they target? Basophils, general inflammation, right? Eosinophils, parasites, fungal infections, and stuff like that. Make sense? Okay? Inflammation. 5 signs of it. Can anyone name 5 signs of inflammation? Or collectively, let's do it. Swelling. No. Heat. Redness. Pain. And the 5th one is loss of function. Okay, so 5 signs of inflammation. Okay. Now, when we talk about steps of inflammatory response, when you have an infection, it causes leukocytosis, so increased production of white blood cells, which then go into the circulation. At the site of injury, they migrate from blood to the tissue extravasate, and following positive chemotaxis, they migrate to the site of injury in the tissue and start phagocytose. Okay, do whatever they're supposed to do, right? Now, antimicrobial proteins. One thing that, let me see, one thing that I'm missing here, actually, and I will write it down. Toll-like receptors. Okay? You don't have to know that, say, TLR5 recognizes flagellin. But you have to understand the concept of toll-like receptor. Remember, we talked about it. Toll-like receptor are pattern recognition receptors, right? That recognize certain molecules on different types of microbes, so-called PAMPs. Okay? Pathogen-associated molecular patterns. Okay? Pathogen-associated molecular patterns. Okay? Toll-like receptors are pattern recognition receptors, PRR. Does that make sense? All right. Yeah, we got through the info. So know what the pump is, know what the PRR is. I'm not gonna throw acronyms at you. I'm gonna throw like the whole name. Like pathogen-associated molecular pattern recognition receptor. All right. Interferons, I think we have about 3 minutes. Interferons, what are the two major types of interferons? Type 1 and type 2. It's just type 1 and type 2 interferons. They just have two types. Production and signaling. What I need you to know is that when interferons produce when they are when cells are infected, when tissue is infected. Okay, so infection triggers interferon production. Interferons are released from infected cells or infected tissue. They can act in autocrine fashion, on the same cell, paracrine fashion on the adjacent cells, and endocrine fashion. Okay? Does that make sense? Endocrine, when interferons go into the blood and deliver to other tissues. The mechanism of defense, they induce production of proteins. Interferon stimulated, they induce the expression of interferon stimulated genes, production of different antiviral, antibacterial proteins. They pretty much warn other cells, right? Remember, understand that mechanism. They don't lyse anything. They don't kill anything. Like they bind to the virus and virus just falls apart. They bind to bacteria and bacteria explodes. Nothing like that. They act on your own cells. Does that make sense? So targets, well, virtually everything. Viruses, bacteria, protozoa.
Hence the name interferon. It interferes with the pathogens.
That's what I believe was the where the name originated from. Yes, that it interferes with replication of the pathogens.
They go to your own cells?
They act on your own cells. They don't act on a pathogen. They act on your own cells so they can mount a response. Okay? Now I'm going to terminate the recording.